Hi, and welcome to another episode of Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Now today, we're continuing on in our series on eukaryotic cells, and this is part 2. We're going to talk about how eukaryotic cells are larger, bigger, more complex than the simpler, but don't call them primitive, prokaryotic cells. Now we all know that eukaryotes are very important, right? And I mean, if you think about this, every plant, animal, and fungus are made up of eukaryotic cells. That's right. There are no multicellular prokaryotes. In fact, all multicellular life on this planet is made up of eukaryotic cells. So, that's interesting to me. We know that eukaryotic cells came from the merger of two prokaryotes. You had this aerobically respiring bacteria, and of course it merged with some archaean. There was maybe a methanogen or something like that. If you think about it, it's pretty wild. A bacteria and archaean merged, and we get eukaryotic cells out of that, with some evolution after that, of course. Now, if you think about this, the defining character of a eukaryotic cell is they have a cell nucleus. Now, the cell nucleus, of course, is a membrane-bound structure around the DNA. And in fact, a few hundred years ago, when people first started discovering cells, they quickly realized they came in two classes. You had your smaller, more structurally simple prokaryotic cells, and then you had your larger, more complex eukaryotic cells. And in fact, the name eukaryote means new kernel. Prokaryote means before the kernel, because you could define the eukaryotic cell by the presence of the cell nucleus. And in fact, today, most people define eukaryotes as the presence of a cell nucleus housing the DNA. And of course, the nuclear envelope, that's a membrane, it's a double membrane structure surrounding the DNA, is pretty wild. It's actually a double membrane, and the reason why is because it's more like flattened sacs stitched together, right, rather than a single membrane. And it does that most likely so it can easily come apart during cell division, and then you put it back together again, so you don't have to take apart your entire membrane. So eukaryotic cells have a cell nucleus. They also have mitochondria, some type of energy converting organelle. Now, of course, we know that mitochondria were once free-living bacteria, and they make the majority of ATP inside of the eukaryotic cell. Now, from an evolutionary point of view, it is actually the mitochondria that are the defining character of eukaryotes. I know we've all been taught that it's the presence of the nucleus, but actually the nucleus evolved after the mitochondria. But because some eukaryotes actually lack mitochondria, they've lost them over evolutionary time. These are parasites that don't need the extra ATP. Some people go, well, not every eukaryotic cell has a mitochondria, but at one point they did. So from an evolutionary perspective, the mitochondria are the true defining character of eukaryotic cells. Now, these energy converting organelles, another one are the chloroplasts. And guess what? Chloroplasts were also once free-living cyanobacteria. So think about this. You got a plant cell, and that plant cell's got both mitochondria and chloroplasts. It's got like two symbionts living within them. Symbionts. They're not true symbionts. They can't, chloroplasts and mitochondria cannot live on their own anymore. They've long since given up that ability, as most of the genes, those original bacteria, have migrated to the nuclear genome. The nuclear genome is just the DNA inside the nucleus. Now, of course, anybody that knows me knows that mitochondria are my favorite organelle. They make, of course, the most majority of ATP. You can keep stacking more and more mitochondria into a cell. That's why eukaryotic cells can get larger, because you can just add more mitochondria. It has nothing to do with surface to volume ratio in terms of diffusion. It does have to do with energy production, and that's important. And of course, mitochondria are involved with their aging process, programmed cell death, which is necessary for growth and development. And like I said, they were crucial in the origin of eukaryotic cells as well, and also involved with the origins of sexual reproduction and two sexes and aging. So mitochondria, I mean, they're pretty amazing. Now, eukaryotic cells, they've got mitochondria, they've got a cell nucleus, and the cell nucleus is actually part of this larger endomembrane system. Now, endo means within, membrane, a system is all these components that work together. So the endomembrane system are this series of organelles that are either connected to each other directly or indirectly that work with inside the cell. And they're involved with the manufacture and breakdown of different cellular components, including molecules and organelles as well. And we're going to come back to this and talk more about this endomembrane system. And then the series after this, I'll talk about the cytoskeleton. Like you and I, we've got a skeleton that gives us shape. And it does more than just give us shape and allows our muscles to attach. It's very dynamic, much like the cytoskeleton of eukaryotic cells. It's a very dynamic system made up of proteins that serve lots of different functions inside the cell, from cell movement to anchoring the organelles to involved with mitosis to actually allowing the cell to move. So the endomembrane system, this is what we're going to talk about mostly today. So it's important for the manufacture and breakdown of molecules and also organelles as well. And it also helps make new membranes. So the endomembrane system is like really, really important. And if you think about this, as a multicellular organism, we've got, not only are we made up of eukaryotic cells that have organelles in them, we also have proteins that are exported outside our cell. So anytime you have a protein that's going to be inside of another organelle, embedded in a membrane, or exported outside the cell, like our saliva, which is something I'm going to come back to, they're made through the endomembrane system. So let's take a closer look here at the endomembrane system. So here we go. The first one, of course, is the nucleus. And the nucleus houses our DNA. So it's like the brains of the operation, right? I mean, this is how it directs all the activities of the cell, and it houses all the information the cell needs for day-to-day basis. Or if you're part of a multicellular organism, it houses all the information to differentiate that cell and for growth and development as well. And the nucleus is surrounded, of course, by the nuclear envelope. Now, attached to the nuclear envelope is the endoplasmic reticulum. Endoplasmic reticulum. I know, more of these giant sciencey words, right? Let's break it down. Endo. Endo means within. Plasmic. Okay, we're talking about the cytoplasm here, the cytosol. So we got this stuff inside the cytoplasm of the cell, and reticulum means reticulate. Oh wait, did I use a word to define a word? Reticulum. It just means like a series of tubes and other things like that. So think of like a branching network of stuff out there. This is the endoplasmic reticulum. And of course, there are two different parts to it. Actually, three. We'll come back to that. But the first one is a smooth ER. It's smooth because under an electron microscope, it looks smooth. And the smooth ER has a couple different functions. And the first one is it's involved with detoxification. So it can break down bad things in our body. Our liver, for example, has lots of smooth ER. So if you're a heavy drinker or you're taking a lot of drugs, what's going to happen is you're going to have all this smooth ER that will help break down these different toxins we take into our bodies. The other thing that the smooth ER does is it helps make lipids. Now, if you remember, lipids are really varied. It could be helping make new membranes. It could be making new sterols, which include cholesterol, testosterone, estrogen, and things like that. Now, there is another type of smooth ER called, oh boy, here it is, Sarcoplasmic Reticulum. I'm not going to talk about that one too much here, but for those of you interested in anatomy and physiology and maybe going on to those classes, the Sarcoplasmic Reticulum, it holds calcium ions used in muscle contraction. Now, the other one is a rough ER. Well, rough ER, you put under a scanning electron microscope and it looks rough. Why does it look rough? It's got these small structures attached to it called ribosomes. Ribosomes. What do these things do? Ah, yes, ribosomes make proteins. So if you've got ribosomes attached to your rough ER, what do you think it's doing? Yeah, it's making proteins, right? So the rough ER is intricately involved with making proteins that will be embedded in membranes inside of other organelles or exported outside the cell. Lots of proteins are exported outside the cell. Another part of the endomembrane system is the Goji apparatus. I often call it just the Goji bodies. And these are involved with the finishing and packaging and transport of proteins. So if you've got a protein that's going to be embedded in a membrane or exported outside the cell, it's going to be made inside the endoplasmic reticulum, okay? And then it's going to be transported by a transport vesicle to the Goji apparatus, and the Goji apparatus is going to put the finishing touches on it, and then it's going to tag that protein where to go. And it can tag it to like go outside the cell, it can tag it to go inside of another membrane, you name it. But that's what the Goji apparatus does. You can think of this thing as like a manufacturing plant. You build your car, right? And then you send it to the Goji apparatus, like you build your protein, and there you put on the finishing touches like the window tinting, the radio, the upholstery, and then you send it out to the dealership. In this case, you send it outside the cell. Now the next one is called a lysosome. Lys means to split. Some means body, okay? So the lysosomes, what they are involved with is breaking down things. So you have a stomach, right? So these organelles are little organs, like our body is divided into all these little things. The lysosomes are like the stomach of the cell. And what they do is they have all these digestive enzymes. You don't want them inside the cytoplasm because it would break down all the proteins and lipids. And lysosomes are also involved with the breakdown of other organelles as well, including the mitochondria. Now I'm going to come back to that point. Think about that. Lysosomes help recycle cellular organelles. And if you're a plant, you have a vacuole. Now a vacuole is a large storage container, and what that vacuole does is it stores water and minerals. This is my friend, and we're in Panama a few years back, and we got some pizza, and we were pretty hungry. We smelled that pizza. Your mouth started to salivate, and your saliva has enzymes in it that start breaking down the food you're eating. And one of those enzymes is called amylase. ACE just lets you know it's an enzyme, and it starts to help break down the starch inside the bread. So you smell pizza, you start salivating, and that saliva's got proteins in it. So how do the proteins that are made inside of a cell get exported outside the cell? So here's how it works. You smell some pizza. Your brain goes, I detect pizza. Your stomach's talking to your brain. Yeah, you're hungry, dude. You need to go eat some pizza. Then your brain sends your signals to the salivary glands. And what happens is inside the nucleus, you have your DNA. And that DNA contains the information to make amylase, the enzyme protein. So it copies it, makes what is called messenger RNA. And then that messenger RNA leaves the nucleus through the nuclear pores out into the cytoplasm. Then that messenger RNA that carries the information to make amylase binds to a ribosome. And then the ribosome, once it binds to it, it doesn't start making the protein immediately. It floats around until it docks to the endoplasmic reticulum. Once it does that, it will begin to take the information in that messenger RNA and translate it into a sequence of amino acids. Now, I'm not converting the messenger RNA into amino acids no more than I convert English into Spanish. I translate the two languages. That's exactly what happens. So we start making our protein inside of our ribosome, okay? We start making the protein inside of our endoplasmic reticulum. Then we form a transport vesicle. We take it out to the Goji apparatus. We put the final touches on our enzyme. Then we tag it. You're going outside the cell. So then it's taken by another transport vesicle outside the cell, where it's then released as saliva into your mouth. I know. Isn't that fascinating? I mean, if you're going, whoa, how does a cell do all of that? Well, people spend their lives trying to figure that out. So that in a nutshell is how the endomembrane system is used to create a protein and export it outside the cell. And as you might have guessed, there's a lot more to this story. So I'd like to end this lecture on, why do we care about this? I think a lot of times we learn something in class and we're like, why did we learn that? Yeah, it's interesting, but what's the real world application? Well, let me just talk about the lysosomes, for example. I'm aging. We all age. I mean, every time I look in the mirror, there's a gray beard and all these lines on my face, they're getting clearer. That's Aerosmith, right? And then, the shoulders start to hurt a little bit. I guess, 30 years of weightlifting will cause that. So we're aging. I feel it every day, right? And I don't want to age. I don't think anybody really wants to age, except I do like the gray beard. I will admit that. But aging is a process. It's really complicated. And there's lots of reasons why we age. And the question becomes is, well, what is that? What exactly is happening as we age? And can we reverse some of this or slow it down? And the answer is, I think so. Let's take the lysosome, for example. Remember, these are like the stomachs of the cell. And one thing that I told you that they did is they do autophagy. Auto means self and phagy means eating. Now, one thing that they do is they also turn over mitochondria. Now, that's important because mitochondria, as you know, are the powerhouses of the cell. So you've seen like little kids and grandparents, right? Yeah, the little kids are running around in circles. My daughter just runs everywhere. My dad sits in his chair. I think he gets tired just watching my daughter run around, right? She's got way more energy than he does. One of the reasons why is because her mitochondria are making lots of ATP. And as we age, what happens is we start to accumulate more and more older mitochondria that become less efficient, that don't make as much ATP. Now, one way that our cells can deal with older, inefficient mitochondria is you eat them. Autophagy. Lysosomes will actually take our organelles like the mitochondria and digest them and break them down. But as we get older, the rate of this autophagy slows down. And the question is, can we reset the clock on autophagy and make our cells act like they're much younger? And the answer is, we think we can. And in fact, they've discovered some chemical signaling inside of our cells that can actually allow these cells to basically reset the clock on autophagy. So this is amazing, right? So by studying one organelle, the lysosome, we can solve one part of a very complex puzzle of aging, and we might be able to live, not necessarily much longer lives, but maybe lives with more quality because we have more energy. And then, of course, as I've talked about many times before with the mitochondria, keep exercising, right? It keeps your mitochondria efficient and keeps your mitochondrial densities higher. So as we age, if we continue to exercise, well, that's great because we will maintain our energy levels and will prevent a lot of the onset of these problems that are associated with aging. So I hope that kind of piqued your interest a little bit in why we might study cellular organelles. Well, until next time, this has been another episode of Tom Kennedy Science.